Welcome, my fellow scientists, to my lecture on cellular respiration. Okay, we're done with it, actually. We're not thinking. Anyways, let's disregard that bit, my fellow scientists, and move on to our main topic. Starting off, do you ever wonder how we get energy from food? Like a sandwich, for example. How does that food come to our body? It allows us to do movement like running, jumping, and even doing a backflip. I can't actually do a backflip. Now, if you've taken a biology course before, you might tell me that the digestive system will swallow, dissolve, and absorb the food that we eat and spread it across our body. But does that really explain how we get energy from food? Not really. In fact, it raises a lot of new questions, doesn't it? I mean, how do we even get food from that? How does that digested form of food turn into energy for our cells? Well, to understand how we get energy, we first need to understand what energy is exactly. In order to understand how we get energy, we need to understand what energy is. And for us humans and most organisms out there, most eukaryotes, it's ATP, which stands for adenosine triphosphate. It's a type of nucleic acid, and as one might be able to guess from its name, has 3 phosphates and adenosine as well as a sugar. These phosphates come in high energy bonds, and whenever you hydrolyze them into ADP and organic phosphate, a lot of energy is released whenever it's broken apart. And that energy can be used in a system or a reaction. Does this seem a little bit difficult to imagine, perhaps to think about? It sounds a bit complicated. To help us, let's imagine a car. But our car isn't moving back or forward since there's no fuel in the engine. And that would be a lot of reactions without ATP. So once we add the fuel, our ATP, we can start moving forward since the spark would be that causes the movement and allows the pistons to go up and down and move the wheels would be the hydrolysis of ATP into ADP and P. That release of energy becomes the spark, if that helps at least. But how exactly do we get that energy in our body? How do we even get to that stage where we have ATP? I mean, it's not like that sandwich we just ate has turned into ATP automatically on its own once it just enters our mouth, because we have to chew it and everything. And that's where cellular respiration comes into play. Now, you might have heard about the mitochondria. Everywhere online, well, not everywhere online, but a lot of times you've heard that the mitochondria is the powerhouse of the cell, and that if the school teaches you anything, that the mitochondria is the powerhouse of the cell. And this is where we can finally learn why. Because most of cellular respiration happens in the mitochondria. To go more specifically, we'll be talking about aerobic cellular respiration, since most humans and most eukaryotes, well, all humans and most eukaryotes use aerobic cellular respiration, and you'll know why at the end of this. Moving on, cellular respiration uses the food that we eat as it comes from our glucose, and the oxygen that we breathe in. Cellular respiration has 4 steps, or, well, 4 processes each, and these processes have many steps. But let's talk about the first 4, the main 4. First is glycolysis, turning our glucose that we got from our carbohydrates into 2-pyruvate, breaking it down. And from there, we're breaking it down further in the pyruvate oxidation process, the second one, the second step, which turns our pyruvate into acetyl-CoA. And from these acetyl-CoA, we can start our citric acid cycle, where we're breaking down our products into usable electron carriers for our last step, which is oxidative phosphorylation, where we're turning all the products that we got into energy. Now, remembering all these names is a bit difficult, but luckily for you, I have Woffick's super awesome mnemonics, where I kind of based it off of the Spider-Man one, the Spider-Man quote, and it goes like this. Instead of glycolysis, pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation, let's remember that great people only create other products. Let's look at our first process. It has 10 steps and 2 phases, the ATP investment phase and the ATP payoff phase. To start, let's understand that this happens outside of the mitochondria in the cytosol, or the fluid that surrounds the cell. From here, let's start with glycolysis, our first step, where it requires a little bit of energy to start. This will be our energy investment phase, where 2 ATP will phosphorylate their phosphate to the glucose molecule, and thus create a fructose molecule, which can break down into a G3P and a DHAP. In the next step, however, that DHAP will also turn into a G3P. Now that we have our 2 G3Ps, we can dehydrogenate them, essentially giving away a proton and electrons to an electron acceptor, which will turn from NAD to NADH. But during this step, oddly, we get some 2 extra phosphate groups that will attach to our G3P, making biphosphoglycerate, if I'm pronouncing that correctly. Now that we have those 2 phosphate groups came in, we now have 2 phosphates on each G3P, which can make a total of 4 ATP, thus paying off the 2 from the beginning, creating a net total of 2 ATP in the end. In the end, not only do we create 2 NADH and a net total of 2 ATP, but we also create water and 2 pyruvate, which allow us to go to the next step, to the mitochondria finally, and start pyruvate oxidation. Now, you've heard me say a lot of things, and they sound pretty complicated, so let's go over some terminology right now. First of all, let's go over phosphorylation. Phosphorylation is the movement of a phosphate group from one molecule to another. This is really important, since it could be the creation of an ATP from ADP and P, as well as the release of energy into a system. An important thing to note is that whenever you are breaking down ATP, it will usually and almost always go into ADP and P. And whenever you are making it, it will usually involve ADP and P. It's a cycle. The hydrolysis of ATP to ADP and P can always be regenerate. And that's important to remember. And that's usually why it's our main source of energy. No, it's made up. Another thing I mentioned was substrate level phosphorylation. Substrate level phosphorylation is the direct combination of ADP and P using an enzyme to mush them together so that they are close together and react and form and create their bond to make a. Another thing I mentioned besides the phosphorylation bit is the term an electron carrier. An electron carrier is as you might expect, a molecule that can take in electrons becoming fully reduced. An example of this would be NAD, which will be reduced to make NADH, or FAD, which can be reduced to make FADH. These molecules will become very useful in the later steps of cellular respiration. As we concluded from the end of glycolysis, our products will be 2,3-carbon pyruvate. That's still left to be broken down. That's still a lot, so we have a long way to go. This step will be turning our pyruvate into acetyl-CoA. It's exceptionally short, but it's really important. Now that we've learned some basic terms, let's move on to pyruvate oxidation, our second step. This one's a bit short, so don't worry. We are turning our 2-carbon pyruvates into acetyl-CoA. Before we get there, our pyruvate will enter into the matrix of our mitochondria, which is the inner fluid, essentially. And from there, it will start to be broken down. We'll start with a decarboxylation, removing a carbon dioxide from one of the pyruvate. Remember, this cycle happens twice since there are 2 pyruvate that are made during glycolysis. The decarboxylation of pyruvate creates some waste carbon dioxide, which we exhale and get removed. After the removal of the carbon dioxide, there is a removal of some hydrogen, which is given to some more electron carriers in NADH. Our final step is adding that CoA to what's left of our pyruvate, making acetyl-CoA. The creation of acetyl-CoA is paramount to the starting of our citric acid cycle, also known as the Krebs cycle, as discovered by Sir Hans Krebs in 1937, and was thus named after him. Let's summarize the citric acid cycle. Starting off, our 2-carbon acetyl-CoA will have their CoA removed, and with the addition of the oxaloacetate, form a molecule called citrate. During the formation of citrate, some carbon and hydrogen as well as oxygen atoms are removed. This forms waste carbon dioxide as well as some NADH. From there, ketoglutarate is made as well, and that turns into glutarate. After glutamate, though, there is an addition of another CoA enzyme making succinyl-CoA. In the process, it extracts another carbon dioxide as well as more NADH. From after we bring in the CoA, we're going to take it away, which produces energy and allows substrate level phosphorylation by turning GTP into ATP. We're almost done. Now that we just have succinyl, we can turn it into succinate after the removal of an electron, and some protons. That makes fumarate, and during the process, it also makes FADH2. Whew, it's a long process. Later, more NADH is made during the oxidation of malate, which allows the reaction to start over again as we finally form the oxaloacetate that goes back into the beginning. In this process, or during one cycle with one acetyl-CoA, we are making 3 NADH, 1 FADH, and 1 ATP. However, this process happens 2 times since there are 2 acetyl-CoAs. So that means we have a total of 6 NADH, 2 FADH2s, and 2 ATP. And all of our glucose that we started with is now broken down, leaving only our oxygen to be dealt with. Before we go over the last step, let's do a little recap of everything we've done so far. All of our glucose has been broken down, but we've only made 4 ATP in total. However, lots of electron carriers in our NADH and FADH2 have also been made. So this last step will be turning all of those NADH and FADH that we've made. We've made 10 NADH and 2 FADH so far, and using them to make ATP. And that's our last step, oxidative phosphorylation. Oxidative phosphorylation is defined as the creation of ATP through indirect measures using the free energy that is created during a redox reaction. There are 2 steps to oxidative phosphorylation in aerobic cellular respiration. The first step is the electron transport chain, and the second step is chemiosmosis. Now, let's start with the electron transport chain. The electron transport chain happens to reside in our inner mitochondrial membrane, where there are 4 protein complexes that control the movement of protons in and out between the mitochondria and its membranes. This starts at complex 4. They all have their NADH and FADH, right? They're filled in. However, an oxygen will pull, since it is highly electronegative, it will pull at our fully reduced NADH that are very stable and have low electronegativity. So they will pull at it, and they have a very strong charge. So it will pull electrons, and these electron carriers now become electronegative. So they'll want to pull on the next set in the complex 3. And that electron carrier will also become electronegative as well. So it'll want to pull on electron 2. And that chain will continue until it reaches the first one, the first protein complex. And during that process, the oxygen will keep getting replaced, so it will keep pulling. That's why it's a chain. And our hydrogens will go up through the protein complexes into the inner membrane, and that will slowly, we'll talk about that after, actually. But how does the movement of electrons really do anything in the long run? As our electron carriers, we are fully stable, right? And they are fully reduced. So whenever we oxidize them and take away their electrons, it releases a lot of free energy in that redox reaction. That energy can be used to move hydrogen through the protein complex and into the membrane above, increasing the concentration gradient. And that's how we'll eventually be making energy. Once we finish our electron transport chain, we'll have moved all of the protons up, the hydrogen protons up to the inner membrane space. Now we can start chemiosmosis. This chemiosmosis is the last step in making ATP. By using the hydrogen that got moved up into the inner membrane space, we are gaining proton motive force. Essentially, since there is a higher concentration above and a lower concentration below, it'll want to move down. But the membrane will not allow it, since it's semi-permeable. Besides the concentration gradient, we're also creating a besides the concentration gradient that we have, we are also creating a electrochemical gradient, since they are charged ions. This means that the inner membrane space is this means that the inner membrane space has a lot of forces that want to pull it back down to balance out and create an equilibrium. However, since, as we mentioned, we're in a semi-permeable membrane, it can't go through. But there is something that will allow it to go through, and that's what creates ATP. That's called an ATP synthase, which allows it to go down through creating forces that make ATP. ATP synthase is sort of like a spinning motor-like organelle that is also resides in the between the membranes. And the movement of the ATP synthase catalyzes a reaction that joins ADP and P, as well as making water. The ATP synthase will go down through it. The ATP synthase spins once with the movement of the hydrogen ions back down. In doing so, it catalyzes a reaction that synthesizes ATP through the combination of ADP and P, which is adenosine diphosphate and inorganic phosphate. An important thing to know about this is that in oxidative phosphorylation, the 10 NADH and the 2 FADH2 reduce to make a total of 34 ATP, with each NADH that gets oxidized making 3 ATP, and each FADH2 makes 2 ATP. From the earlier reaction, you can tell that we have a total of 38. However, this depends on whether or not some ATP is lost moving through the cytosol to the mitochondria. Our ATP totals are as follows. Aerobic cellular respiration creates 38 ATP, around there. This can go down to as low as 32 ATP, depending on whether or not energy is lost moving through to the mitochondria. Or about 3,000 kilojoules of energy. However, a lot of this comes from the last process. Glycolysis only makes 2 ATP, pyruvate oxidation makes 0, the citric acid cycle makes a total of 2 ATP, and oxidative phosphorylation makes 34. The importance of it all. With our 38 ATP created, we're making so much more energy than anaerobic cellular respiration, since it only creates about 20 or a 20th of the amount of energy, which is around 2 ATP, since it can only do glycolysis. And as we are a bit more complex, we require so much more energy, since and so this 38 ATP is well appreciated. This ATP can be used for almost anything, whether it be neural signals, muscle movements. Thankfully, we our body is very appreciative of all this ATP that we create, since we can use ATP for movement, neural transmission, and the making of DNA. On a daily basis, our bodies require around 100 to 150 moles of ATP, which totals to around the average body weight, which is 60 to 70 kilograms. That's a lot. Luckily, we have 30 trillion human cells in our body, and most of them can make ATP. Let's look at some ATP at work in our bodies. Specifically, let's look at neurotransmission. The human brain requires a lot of ATP, up to 25% of the amount of energy that we use a day. Some studies suggest that it requires about 5.5 kilograms of ATP for a resting brain for just a single day. And only one neuron would require billions of ATP per second. Neurotransmission is defined as sending signals between neurons to neurons and other receptors. It is highly important for our body, since it can control our mind, our muscles, and everything in between. ATP plays a vital role in controlling neurotransmission. In order not to go too in-depth on the process of neurotransmission, since it is quite long, we're just going to do a short summary and how ATP affects this process. Neurotransmission contains 4 steps. Starting with step 1 is sending an action potential or an electrical impulse down our neuron's axon to a presynaptic terminal. The next step is chemical release. The second step is a very important one, where the calcium ions that have come in from the calcium channels that we just opened, they will rush into our terminal. These calcium ions will lead vesicles containing neurotransmitters to exit our neuron through the cell membrane in a process called exocytosis. However, these neurotransmitters are not out of the clear, as they need to diffuse to the other side of the synaptic cleft, or a little gap between the next neuron. After the neurotransmitters have been dumped and diffused to the other side, they need to bind to specific receptors, where it will cause an opening or closing of ion channels, which can increase the chance of a local action potential in the neuron that it's now in, or it can inhibit the action potential by reducing the chance that it will cause another impulse to happen in this new neuron. And that leads to the final step. this leads us to our last step, which is essentially the cleanup, where the neurotransmitters are quickly removed from our receptors in order to stop the signal and reset to receive another. You might be asking, where exactly does ATP come into all of this? Well, it comes into two parts of neurotransmission, starting with the first one, which is resetting, where we send our vesicles across to the next neuron. But before that, in order to pack the neurotransmitters inside of the vesicles, it requires ATP to do so. And in order to send those vesicles and release them in the process of exocytosis, that also requires ATP. So much so that there's even mitochondria inside of those neuron terminals. The second part that requires ATP is resetting after an action potential. Since an action potential requires a change in ion concentration, since originally it started as negative, but once it opens, more positive ions come in, increasing the voltage, and that voltage change is the action potential. But once it passes, it needs to go back, so that way another action potential can come through and send a signal. So it's up to the ATP channels or ATPase to reset and imbalance our neurons to make them negative once again. It takes approximately 1 billion ATP to send one action signal. Thankfully, we have our sodium-potassium ATP pumps, which use active transport to reset the imbalance to make it negative once again. That concludes my lesson on cellular respiration. I hope you have a good day, and good luck on your next test.